In our previous episode, we discussed about the mechanisms for how light enters through your eye and get focused on a point on the retina, right? Now let's discuss it more precisely with utmost details. There are many ways through which animals process visual input. These inputs are based on photoreceptors on our retina. Photoreceptors are specialized cells for detecting light. The retina consists of millions of cells packed together in a tightly knit network spreads over the surface of back of eye. These cells can be divided into three basic cell types: photoreceptor cells, neuronal cells, and glial cells. Now, photoreceptor cells consist principally of rods and cones. Rods are responsible for vision at low light levels, also called as scotopic vision, while cones are responsible for vision at higher light levels, called as photopic vision. The quality of vision provided by photoreceptors varies enormously among animals. For example, mantis shrimp have 16 types of photoreceptors, unlike humans who only have 4, which enables them to see in the wide ultraviolet and infrared regions and even polarized light. It's fascinating, right? On the same technique, humans devised night vision and thermal vision goggles. While night vision devices use optoelectric image enhancement, which works by sensing small amounts of infrared light reflected off objects and then electrically amplifying that light into a characteristic glowing green image. Thermal vision, however, works by detecting infrared radiation from the relative temperature of objects around us to help us see warmer objects, like people and animals, against cooler backdrops. In humans, rods have spectral sensitivity that differs from cones. In other words, rods do not mediate color vision, for that's why we only see black and white world in dim light. Cones give us color vision. Cones are concentrated in the center of our retina in an area called the macula and help us see fine details. The retina has approximately 120 million rods and 6 million cones. Despite all these, humans are not created for excellent night vision. Comparing a human eye with an owl, which has 5 times more rod density and multiple biological reflector systems, serving as a second opportunity for enhancing light sensitivity among photoreceptors. For example, some vertebrates have a layer of tissue behind retina called tapetum lucidum, enabling them to see and even pray at extremely lower light levels. Nevertheless, humans are adapted to dim light scenarios more perceptively, as they have higher brain functions performing simultaneously to assess the situation. Higher IQ have some own perks. Now let's dive deep in and know the exact physiology happening in your retinal cells. For this, you have to know the light first. Light is electromagnetic radiation with wavelength between 380 to 750 nanometer. Light travels in form of small energy packets or particles called photons. Now let's assume you are on your rooftop and you saw some red fireworks. Red color has wavelength of about 650 nanometer. Light enters through your eye and penetrates the cellular layer of your retina. This light falls onto rods and cones. As we know, red is a specific color signal comprising of specific wavelength. It will be detected by cones. There are 3 types of cone cells: red sensing cones, green sensing cones, and blue sensing cones. When a cone cell is hit by a right colored photon, it excites and sends an electrical signal to the brain. In the cone cell, there is a pigment called retinol. Retinol is held tight with a protein called opsin. Different photopsins are sensitive to light of different wavelength. Retinol and opsin are joined together by double bonds. When a right-colored photon of specific wavelength hits them, light energy is absorbed by it and double bond is converted to single bond. For a split second, it releases an electron enough to kickstart an electrical signal. This electrical signal travels towards the bipolar cells. Bipolar cells exist between photoreceptors and ganglion cells, behaving like a bridge. They act directly or indirectly to transmit signals from the photoreceptors to the ganglion cells. Ganglion cells are the final output neurons of the vertebrate retina. Ganglion cells collect information about the visual world from bipolar cells. Ganglion cells are also the most complex information processing systems in the vertebrate retina. There is a circular area at the back of your retina known as optic disc. It's the point where the axons of the retinal ganglionic cells come together. In other words, where the optic nerve connects to the retina. Because there are no rods and cones, it is also referred as blind spot. Different subsets of ganglion cells register different types of visual information. For instance, some ganglion cells are sensitive to the contrast in movement, others to shape and details. Together, they carry all the necessary information from our visual field. The brain allows us to see in 3D, giving us depth perception by comparing these signals from both eyes. The signal generated in the retina end up in the visual cortex, a part of brain that is specialized for processing visual information. Here, the impulses are stitched together to create visual images. In our upcoming episode, we'll go over this in more detail. Stay tuned and keep following sqadia.com for more fascinating medical videos.